Okay, this is part 4 of the chemistry slash macromolecule lecture. Last part, we talked about how all macromolecules are both made and broken down, dehydration, synthesis, and hydrolysis, respectively. The rest of this lecture then just focuses on each of those 4 macromolecules. So the first one is going to be the carbohydrates. As we go through this, you just want to focus on its functions, examples, those sorts of things. So for these carbohydrates, they contain carbon, hydrogen, and oxygen, and its major function is to supply a source of cellular food. So really, we're talking about short-term energy. And just make sure that you understand that it is short-term energy. Okay? The building blocks, which we're going to identify for most of these macromolecules, are called saccharides. You can also call them monosaccharides. So now what we're going to do is we're going to take a look at monosaccharides, disaccharides, and polysaccharides. So for the monosaccharides, they usually have 3 to 8 carbons and are named according to the carbons. In biological systems, the 3 that predominate that we see the most of are going to be a triose, which is 3 carbon, a pentose, which is a 5 carbon, hexose, which is a 6 carbon. So remember that triose is 3, pentose is 5 carbon, hexose is 6 carbon. Now what we also have then is with monosaccharides, they will always have a carbon to hydrogen to oxygen ratio of 1 to 2 to 1. So what that tells us then, if I say we have a triose, you say, what is the chemical formula of a triose? Well, you know that it has 3 carbons. If it has 3 carbons, it has to have double the amount of hydrogens, right? Because we're going 1 to 2 to 1. And then the same number of oxygens, right? So then a pentose, has 5 carbons, so then it has to have double, and then for oxygen, it has to have the same number, okay? And then a hexose would be C6H12O6. Examples, glucose. I think all of us have heard of glucose. We have blood sugar or blood glucose levels. Almost every single cell on this planet utilizes glucose for energy. Fructose is a sugar found in fruit, and ribose is a sugar found in nucleic acids. So what I want you to know in addition is that ribose is a pentose, and glucose and fructose are hexoses. So glucose and fructose have 6 carbons, ribose has 5 carbons. All right, so that's it for the monosaccharides. Disaccharides, they're made up of 2 monos that are going to be joined together via dehydration synthesis, which we already know. And then you just have to know examples. So you do have to know these. So sucrose is made up of glucose and fructose, and that's the sugar that you add to your coffee. Lactose is made up of glucose and another monosaccharide that we haven't covered. You don't have to worry about it other than knowing that glucose and galactose make up lactose, and that's a dairy sugar. You may have heard of people that are lactose intolerant. They lack the enzyme that recognizes this union, so it can't break that down. And maltose is malt sugar, sugar found in some beer, and that is made up of glucose and glucose. Should I cover that now? Yeah. Okay, so we know that glucose is C6H12O6, and fructose is C6H12O6. But sucrose is not C12H24O12, and that's because they're made together via dehydration synthesis. So we have to subtract a water from that. Now, out of this water, the most important thing is going to be this oxygen. So what that means is the larger the carbohydrate, the effect of oxygen becomes less and less. So a monosaccharide is going to be more polar than a polysaccharide, right? Because as we keep adding these monosaccharides to it, we keep losing water and we keep losing that oxygen. And if you remember from chemistry, that oxygen is extremely electronegative. Sorry, I'm just going to try and shut this. Okay, so it is the larger the carbohydrate, the effect of oxygen becomes less and less, and that's the important part. So when we go to these polysaccharides, we're going to be seeing this part right here. They're less soluble in water, right? Because if something is polar, we know this from going over the properties of water, it will be water soluble. So we're not saying that these polysaccharides are nonpolar, we're just saying that they're less polar. So if I said which of the following is most polar, and I gave you some examples of, let's say, glucose, I wouldn't do fructose, sucrose, and starch, you would then know that glucose is the most soluble in water. Right? Hope that made sense. So now that's all that's left for the polysaccharides is to go over examples. Okay, glycogen is a polymer of glucose. Saying that it's a polymer of glucose just means it is made up of glucose, all of these glucose molecules strung together. It's a storage form of energy used by humans. It is predominantly found in the liver and muscle. Starch is also a polymer of glucose, and it's the storage form used by plants. And then finally we have cellulose. It's also a polymer of glucose. It is found in plants, right? It makes up the cell wall of plants. So why are we talking about it in human anatomy and physiology? Well, cellulose is not a good source of energy. It cannot be hydrolyzed. Hopefully that word sounds familiar. Hydrolysis, right? That's the reaction by which we break down these polysaccharides. So cellulose cannot be broken down into glucose. So even though it's made up of glucose, it has a unique linkage, and we do not have enzymes that recognize that linkage between these glucose molecules. So we can't break that bond, so we can't hydrolyze it for energy. So we call that fiber. It just goes through our digestive tract undigested. Okay? And then I have one image. It's more just to kind of show you guys. So here's glycogen. Here is one glucose, another one, so on and so forth. Okay? And this is the linkage for cellulose that I was talking about. It has a unique linkage. It's different than this. And again, we don't have enzymes that recognize that. And that's it for part 4, I believe. Yeah, part 4.